restoration and habitat modeling of the Rocky Shoals Spiderlily. Our population in Stevens Creek. He has worked for the U.S. He's got a pretty wide range of work that he's done. He's worked with the U.S. Fish and Wildlife Service at various wildlife refuges across the country. After graduation, he hopes to become a refuge manager, a biologist with the U.S. Fish and Wildlife Service. We're helping to put a really good well-trained person out there. He has been working for about a year and a half, and thanks to some degree to a grant that the Native Plant Society supplied him on the various aspects of Rocky Shoals spider lilies, which he will explain to you. And he's proposing to present his final thesis research next spring. Andy, go to it.
All right, thanks, Bill. And thanks everyone for coming out to hear about my research, and for all of you who contributed, well, that's everybody, to my grant. That's been a huge part of why I've been able to do this. So let me share my screen here. All righty, is everyone able to see that?
Yep.
Yeah.
No.
Try it again. Let's see here. Is that any better for anybody?
It's visible.
Yes.
So.
Looks good.
Okay. Well, as Bill said, my research is revolving on the restoration habitat characteristics of Hymenocallis cornaria in Stevens Creek, South Carolina. And I just want to mention my committee, which consists of Althea Hagan, who's my advisor, and Mark Scott and Kyle Barrett, which are both professors within the Department of Forestry and Environmental Conservation. And Mark also holds a dual appointment with the Wisconsin DNR. So a little background about Hymenocallis cornaria. It's commonly known as the Rocky Shoals spider lily, at least in South Carolina. In other parts of the country of the Southeast, it can also is known as the Cahaba lily or the Shoals lily, or sometimes just the spider lily. But in South Carolina, people tend to call it the Rocky Shoals spider lily. It's a perennial bulbous, perennial bulbous and emergent macrophyte that is known for its large, beautiful blooms. It's endemic to shoals of fall-line streams in Alabama, Georgia, and South Carolina. And unfortunately, H. cornaria has been declining across its range due to a variety of things, including dam construction, water quality degradation, and herbivory impacts. It was originally put up for endangered species listing in the late 1980s, but it was ultimately rejected. But it is listed as rare and imperiled, and is a high priority within the State Wildlife Action Plan for South Carolina. So a little background about shoals, because that's kind of the most important part about spider lilies. So in our part of the country, shoals refer to fall-line geology, which creates bedrock outcroppings that transect streams, causing a series of waterfalls, rapids, and exposed bedrock. So these shoals tend to have shallow water depths, high stream gradients, and coarse, patchy substrates that support high biodiversity, which Stevens Creek is definitely an example of that. And H. cornaria itself has a unique and shoal-dependent life history in which their plants grow directly out of the shoals, flowering in May and June. Their large green photosynthetic seeds get wedged in shoal crevices by stream flows, producing leaves, a bulb, and contractile roots that keep them anchored to the shoal substrate. Under ideal conditions, they grow in large clonal clumps to the production of ramets and seed deposition within the mother plant, which forms large colonies that can cover entire shoals. As you can see here, the picture on top is a uninhabited shoal in terms of spider lilies, and then here is the Parks Mill colony with the lilies covering up almost all the available space for that. All the available shoal space. So the history of scientific literature on the Rocky Shoals spider lily is pretty narrow, but I have a quick summary of it here for you to get a little background. So Larry Davenport was the first to really study this species in detail, and he studied mostly the botanical characteristics and did a demographic study, finding out where it is, where it isn't. In 2003, Smith and Garland did a taxonomic study on this Hymenocallis genus and formally declared Hymenocallis coronaria its own species. And that was actually one of the reasons Fish and Wildlife Service initially denied Hymenocallis coronaria for endangered species listings, because they thought it was too genetically similar to other Hymenocallises. But Smith and Garland disagreed with that. Then in the late 2000s, a guy named Scott Markworth and various colleagues did a lot of work with the genetics and distribution of the spider lily. And their big finding was that genetically, the species is split down to an eastern distribution and a western distribution. So we are in the eastern distribution of the spider lily. So it basically is down the middle of Georgia. So western Georgia and Alabama is a western, what they call haplotype. And in South Carolina and eastern Georgia, it's they call the eastern haplotype. But something interesting that they also found in this paper is that our colony of spider lilies on Stevens Creek is actually more similar to the western haplotype and doesn't appear to fit in with other spider lily genetics within the Savannah River Basin. So that's pretty interesting. We'll talk a little bit more about that later on. And then in 2007, a lady by the name of Cindy Albach Smith did a survey of Stevens Creek for a potential quarry installation. And she was doing it for basically for mitigation purposes and to see how big of an effect the dam would have, or the quarry would have on the lilies. But from that, she came up with a lot of good information on the current distribution and ecology of the lily within Stevens Creek. So I've spoke with her a lot and built on off her research. And then also in our neck of the woods, Judy Gordon and I'm blanking on Ware's first name, but Gordon and Ware did a study in the Savannah River Basin comparing a colony in the Augusta Shoals with our Stevens Creek population, and came up with some conclusions, but mostly qualitative in nature. And then in 2014, there was a pollination study at Lansford Canal on the Catawba River in South Carolina, looking at what kind of pollinators come and visit this species. And they originally thought that it was going to be a specific type of hawk moth that had to pollinate it and only pollinate it at night. But what they found out is that lots of things pollinate it both during the day and during the night. And if I'm not opposed, if anyone has any burning questions, if they want to interrupt me while I'm going. Otherwise, I'm planning for lots of time for questions at the end. So based on that past scientific literature, me and my advisor and my committee started to form some questions. What are the research gaps? What don't we know about this lily? And one thing that immediately jumped out was restoration. So there's been a lot of interest in doing restoration of this species, especially in the last 10 to 15 years, but there's been zero published research about it. And any past attempts have mostly ended in failure or poor documentation. So besides the need to learn about how to do restoration, there's quite a bit of impetus to do so. So it has various important functions within a stream ecosystem, including nutrient cycling, its importance to food webs, and provides really developed habitat structure. It's also a culturally significant species and an indicator species. A lot of people in the Southeast view the spider lily as an indicator of free-flowing, high-quality streams. So if you have spider lilies, you have a good quality stream. And there's also quite a bit of public interest in blooms. If you've ever been out, get a chance to get out to, well, first of all, our site on Stevens Creek, but this picture here is from Lansford Canal on the Catawba River at Lansford Canal State Park. And the ecotourism around this park while they bloom is really, truly incredible. Lots of money flowing into the park and into the state to see these blooms. And I've actually helped out with a restoration project on the Chattahoochee River in western Georgia, where a rafting company is funding this nursery to restore lilies on the Chattahoochee so they can do special rafting trips to show people lilies. So there's some money to be had. And then, of course, there's been interest from you all, South Carolina Native Plant Society, and landowners in the Stevens Creek area in restoring and increasing the amount of lilies on the creek. So then the other major research gap we noticed was that these habitat characteristics, every time you read about the lilies, they talk about these things. So you need to have shallow and swift flowing water, you need an open canopy, you need clean and clear water, and you need these bedrock crevices or coarse cobble substrate for lilies to establish in. But nobody's ever put these descriptions to numbers, like what is shallow water? How swift is too swift? Do they really need open canopy? What water quality parameters is most important? What size of substrate is most important? So that's where my second question came in. So we need to better understand these habitat characteristics to inform conservation and restoration decisions. So that leads to my research objectives, which the first objective is to examine the effects of technique, life stage, and season on Hymenocallis cornearia restoration, with special attention given to efficiency and replicability. And then my second objective is to identify and quantify habitat characteristics for H. cornearia establishment at the microhabitat and shoal scale. So we'll get into how I'm working on those objectives and what I've learned so far later on. But first, let's well, Stevens Creek isn't really even in the upstate. But so here you can see where Clemson and Anderson is. You can see a little airplane for Greenville up here. Upper Stevens Creek flows into the Savannah River just above Augusta. And where our site is not too far away from the confluence of Hard Labor and Cuffytown Creek, which form Upper Stevens Creek. So I don't have it overlaid on here, but if you can imagine a triangle kind of from Greenwood over and then down to where that pin is, that's kind of the watershed of Upper Stevens Creek. And Stevens Creek is known for its high biological diversity. It's one of the most biodiverse aquatic systems in South Carolina and was listed as one of the top 15 southeastern streams to target for aquatic conservation. And if you ever get a chance to get down there, or if you know how great it is, you know what I'm talking about. It's an awesome place. So if we get into my study stream segment a little more in detail, pictured here to the right is the 8 kilometers of Stevens Creek that I'm working in. It's near a small town of Plum Branch, South Carolina, which is fairly close to McCormick, South Carolina, which if you're not from the area, there's not a whole lot down there, which is primarily why it's so great to study the spiderlilies in this area. So there is very little development. The land use is primarily pine plantation. And as Bill mentioned, there is a wastewater treatment plant up in McCormick, but we'll talk a little more about that later. But it appears that they've made some improvements to their discharge protocols. It doesn't seem to be too large of an issue, relatively speaking. So it's something else really unique about this section of Stevens Creek. It has an extremely high density of shoals, many of which support lily populations, some do not. If you kayak the creek or see the creek above or below this stretch of creek, there are very few shoals, but in this section, there's somewhere around 25 to 30 unique shoal units. So that's pretty unique and a great place to study study the shoals. And then so if we're looking at this map, you can see the blue dot is the Parks Mill site. This is the site of the South Carolina Native Plant Society Preservation Project. There's the non-functional turn of the century grist mill and its associated diversion dam, which is not owned by South Carolina Native Plant Society, I must add. I don't want to confuse that. The dam is important. The dam is fully intact and still traps sediment and modifies flow to some extent. And I hypothesize that the dam has something to do with why the lilies at Parks Mill are doing so well, but I have yet to quantify that exactly. So then besides Parks Mill, my other 2 main study sites are this red dot here and then the orange dot. Those are both interested landowners, one of which who's been involved was interested in having his site restored. He had a lily colony there for a long time, and within the last 15 to 20 years, it has declined compared to its historic extent. But since I've been around, it's been rebounding, and that's not just because I've been planting lilies there. There's been lilies that I have not put there that have popped up since in the last couple of years. So that's good to know. And then the middle site is a low density shoal. There's actually just one clump of lilies in that site. So we kind of have a high, medium, and low sites for our study. So jumping into the restoration methods. So I'm assessing restoration success in experimental plots with different treatment combinations. I'm also looking at how many person hours were needed for the purposes of a time-based cost-benefit analysis. So how efficient is it to do this restoration work, and how replicable is it? Because as much as we all love plants, too much effort for one restored plant may not be worth it to people, or may not be worth it to people who want to fund it. So to do this restoration, I collected seeds from the Parks Mill colony in 2020 and 2021. 2020 seeds were used almost entirely for bulb propagation, and 2021 seeds were used for seed and seedling based treatments. Seedlings and bulbs were grown hydroponically in Clemson's greenhouse, pictured here, which made up a large portion of the person hour effort. I had a lot of undergraduate students working for me, and they were able to help quite a bit with that propagation work, but a lot of hands and a lot of time was necessary to put this all together, especially at the very beginning. Me and my first summer's undergraduate technician spent a lot of time getting all these lilies potted up. So speaking of that hydroponic method, I'd be remiss if I didn't mention the importance of Joe Townsend for the development of my propagation methods. And I was very sad to hear of his passing, but I'll always be thankful for his help. I'm sure he'd be very happy to see the amount of lilies growing in the creek thanks to his tips. So my greenhouse setup consisted of kiddie pools with 2 to 4 inches of water, aerated by aquarium pumps and air stones. And these seeds were placed in pools and aerated until they germinated and developed a radical, hypocotyl, and cotyledon, which I define as the seedling stage. We'll see some better pictures of that coming up here. So once they developed those key plant parts, they were potted in standard greenhouse pots and filled with approximately 3 inches of locally sourced surge rock and 5 inches of sand. And then they were grown in our greenhouse until they were outplanted in the creek, the last of which went out this past July. And a little side note, undergraduate research found out or determined that there was not any statistically significant difference between using an aquarium pump versus using an air stone versus honestly using no aeration at all. These seeds really just want to germinate, and they germinate almost 100%. So that's nice to know. Into visual representation. So here are the green seeds at about time of harvest. These seeds are photosynthetic. So this green outer part is photosynthetic, and it will grow the plant parts right out of it with the seed still attached. So here we can see when potted in with the shoot coming out, and then below that is a little bulblet and little roots coming out. And we'll see more pictures of that coming up. But this is what it looked like after they germinated and went into the pot. Here is a bunch of seeds getting aerated for germination. And here is a bunch of young seedlings with diatomaceous earth spread around them because we had a little cutworm infestation, and cutworms were getting in there and slicing off the top of the lily stems. So going into techniques that I use. So there's primarily 3 techniques I use to restore spider lilies. The first one is the wedge technique, which you can see here is exactly what it sounds like. All these examples are with bulbs, but taking a bulb or any propagule for that matter, and just finding a place in the shoal area and just shoving it in there and hoping it stays there. And you can move rocks to put it on top of it, lift rocks up. Little crevices in the bedrock like here work really well. but there's definitely a limited amount of those around. The second technique is a Coir Wrap, which was adapted from a method Joe had done in the past with a natural land trust project. But they use plastic materials and I adapted to use coir, which is a biodegradable fiber made from coconut husks. So what's going on inside this wrap is there's a large piece of cobble inside here that you can't see. And then all the lily bulbs or whatever propagule we used is put into this wrap or put on top of the cobble. And then we wrap the coir matting around it and thread the stems through the holes. And we'll talk more about how successful that was later. And then we tied up the sides with more coir. So this is kind of like, as my advisor calls them, this is like an empanada. That's the casual name, unofficial name for what these are. It's an empanada technique. And then here we have a coir mat, which is kind of hard to see, but it's exactly what it sounds like. So we threaded the stems through the holes of the coir and laid the coir down on as flat a spot as possible as we could find in the creek, or as suitable a spot as possible. And then we put whatever size cobble we could safely lift on top of it. So those are the techniques. Oh, sorry, not pictured is the broadcast technique, which, as you can imagine, involved throwing seeds at random into a shoal. This technique was not included in the overall analyses due to the differences in plot size, issues with assessment feasibility, and lack of treatment replication. But we will include it in our analyses of efficiency because it's very efficient. So then moving on to our life stages. So these are the 3 life stages we used. The first one here is a seed. So this is how a seed looks pretty much right after being and restoration efforts almost immediately after. So they could sit in a pool for a while getting aerated, but some seeds we collected the day of and used them in restoration that same day. And then here's a seedling. So this is once it germinates, these plant parts come right out of here. Here are the young roots, little bulblets, beginnings of a little bulb, and then the first shoot. So this is when we would pot them in the pots. If we were going to grow them into bulbs, or if we were going to use them as seedlings in a seedling-based treatment, we used them just like this. So the bulb and all plant parts will keep growing for about 2 to 3 months before the seed actually shrivels up and dies or decomposes. So and it stays photosynthetic for that entire time. So when we plant these seedlings, we plant them with the seed still attached into a spot. And then the last one is, as you can see, the bulb. This bulb is about 6 months old. I think this is a spring picture. Well, actually, that would be more like 8 months old. So you can see how developed these roots are. There's the thicker roots, which I don't know how well you can see, are the contractile roots, and they literally contract and like cement the bulb into the creek. And then below that are the more fibrous roots. And then this one, you can't see, but there's a second little stem coming out the side. But mostly fall and spring efforts, they didn't have more than one stem, but in the summer they had multiple stems. Moving on to the last kind of factor I'm most interested in for restoration, and that's seasons. So in fall 2020, we outplanted bulbs into shoal 3. And in spring 2021, we outplanted more bulbs into shoal 3. And then this summer, August of 2021, we did all life stages in shoals 1, 2, and 3. And the reason we didn't do seeds or seedlings in the fall and the spring is because it doesn't work for their life history. Seeds will become seedlings whether you want them to or not. You can't freeze them; it'll kill them. They'll become seedlings by the time you want to use them. So the only time we could do seedlings and seeds is when the plants are naturally producing those life stages. And then the broadcast treatment was also only done in summer because that's the only time we had seeds. So each season consists of 3 wedge plots and 6 to 8 plots each of Coir Wrap and mat. So we had those replications each season. And here is the map. Here is aerial imagery map made in GIS of a normal setup. This is from the fall. So these orange plots are 10 by 10 meter wedge plots. So we planted usually around 28 to 32 lilies within each one of these 10 by 10 plots. And then each one of the purple and blue dots represents a Coir Wrap or a Coir mat. Moving on to results. So looking at the results overall, we planted 843 Hymenocallis coronaria individuals into the 3 shoals on Stevens Creek, and that does not include the broadcasted seeds. Of those 843 individuals, we had 218 individuals establish themselves, and here just, which is a 25.9% success rate, which I don't know actual numbers from anyone else because nobody's published or shares their information, but I think that's significantly higher magnitudes of anyone else's success. When I was helping out in the Chattahoochee, they were happy just to get like 1 or 2. They could remember all the lilies that they actually got established. And obviously at this point, I don't remember all 218 by heart or by name or anything like that. But here are just some pictures of examples of established lilies. Here's a wedge one, wedged in between some rocks. This was a spring restoration effort. And then here is a wrap from this past summer. You can kind of see the remnants of the wrap there still. One note about the coir is it's supposed to not biodegrade for up to a year or 2, 3 to 4 years if not getting wet, but these are obviously getting wet. So we've seen that they last at least a year. The ones from last fall have at least lasted, some of them until now. But eventually they're going to biodegrade, and the lily, in theory, has established itself by then. So if we break down the restoration results by the factors I'm most interested in, we'll start with technique. So this is lumping all the different life stages and all the different seasons together and just looking at what we had for all the different techniques. So we've found that wedge had a 31.7% success rate, the wrap 28.9% success rate, and the mat only 12.4% success rate. And then the stars indicate statistical significance using a chi-squared analysis of means proportions. So the wedge, what that means is the wedge had a significantly higher overall mean, or significantly higher than the overall mean proportion of success, which if you remember is about 26%. And the mat had a significantly lower success rate than the overall mean proportion of success. If we jump ahead to looking at lumping all the seasons and techniques together and just looking at how we did life stage wise, the seeds had 18.6% success. Seedlings had 32.4% success, which was significantly higher than the overall mean proportion of success. And then bulbs had a 25.5% success, which is pretty much right about the overall mean success rate. If we look at the restoration results by season, you can see that fall did not have a very good success rate, looking at 9.9%, spring 26.2%, and summer 32.1%. So the summer was significantly higher than the mean proportion of success, and the fall was significantly lower. Looking at restoration efficiency, so how replicable is this? Because not everyone has, let's go back here. Not everyone has all the time I have, not that I have tons of time, but to do this. And as you can see, there was a significant amount of effort put into this, and the vast majority of it comes from the propagation time. So if this table represents just the person hours needed for the bulb-based techniques, depending on the season. So you can see just to propagate bulbs to use in the fall required 73.75 person hours. And I guess I should explain, a person hour is the amount of hours it took to do something multiplied by how many people there were. So I just realized there was a chat. I can go back to that, Ryan, or do you want me to address it at the end? I didn't notice that till now. Failure of mat treatments.
Yeah, you can catch it at the end. It was just a thought.
Yeah, I'm going to touch on some reasons for different success and failures at the end. That's a good question, though. So a person hour is what I was saying is the amount of hours it takes to do something multiplied by the number of people that did it. So if it was me in an undergrad, worked 4 hours, 2 times 4 is 8 person hours. So if you look at propagating bulbs for the fall, that took 74 hours or about. If you want to keep those bulbs growing and alive over the winter to the spring, that takes 103 hours. So that's total. So another 30 hours roughly. And then if you want to keep them alive until the summer and ready to go, that takes another like 26 hours, person hours that is. Then you can see the actual techniques themselves pretty much a wash in terms of how much time it takes. The mats tend to take the least amount of time, and the wraps tend to take the most, especially the first year. But we were able to improve the efficiency of the wrap technique by bringing the cobble back to the greenhouse and assembling them ahead of the restoration effort. And the time it takes to do the wedge treatment is largely dependent on how picky the planter is when choosing wedge locations and how dedicated and fastidious they are to wedging the lily in there. I've seen some undergrads do 6 lilies in the time it took me to do 1 because they aren't as committed to seeing them survive. And I try to get them to put the necessary effort in, but definitely sometimes wedge can take longer if you're really trying to get the lilies into good spots, which this summer we really did, and you saw that success was really high. Moving on to looking at comparing just restoration results in summer of 2021. So, and the reason we're looking at that is because we have all the life stages to choose from, and you can really start to parse out some relationships. So if we look at just bulbs, you see that we had a 43.5% success rate with bulb establishment in the summer. And within that, wedge was 32.1, wrap 80%, and mat 30. So those are all pretty good numbers, especially that wrap number. But as you know, keeping the lilies alive until summer requires that additional effort. But what doesn't take effort, look at these person hours, 149, 138, like those, that takes a lot of time. But seedlings does not take very much effort. And we found overall seedlings had 34% establishment success in the summer. Specifically, wedge had 49% success for seedlings. That's a pretty good number, considering it only took 16 hours to harvest the seeds, germinate them into seedlings, and then actually wedge them out there. And the wrap and mat, pretty good success, but not that great. We're looking at seeds, overall only 18.6%, and the wedge and wrap had pretty good success, and then mat with seeds and not work very well. So a couple little, I'll take opportunity here to do a little bit of inference on why that happened. The mats in general, once again, it depends on your effort level. It takes a lot of work to move mats or to move rocks to put on top of the mats. And depending on the supervision level and how dedicated everyone is, some of these mats weren't weighed down very well, or it just wasn't possible based on the cobble we had around. So definitely fall mats that had to deal with winter stream flows the whole time, definitely almost all got washed away. I don't think we had any mats successful from the fall, and we barely had any wraps either. I think we maybe had 1 or 2 wrap lilies established, but it was primarily wedging from the fall. And if you think about the wrap treatment also, I had a professor in grad can move that rock easier. So basically, when you're putting something into the stream, a rock, you have to expect that it might move. And so some of these wraps or mats may exist somewhere, well, not the mats, but the wraps may exist somewhere, but they're no longer in our plots where we're searching for them. And that's where the wedge comes in. The lily, the way the life history of the lily works, they really need to wedge in with these contractile roots. So that's why my personal opinion is that from my data, not just my opinion, is that the wedging is the best because it gives them a chance for the lily, whether it's a seed, a seedling, or a bulb, to do what it's supposed to do based on its life history. The roots have to fend for themselves and weasel into little crevices and latch on. When you put them on a wrap or a mat, they kind of have, which is what we were testing, but they get this support system, but then are they going to attach to that support system, or are they going to attach to the actual creek because of, or the actual substrate? Because eventually that coir is going to break down, and we don't know if they're going to continue to stay there or not, but we're going to continue to keep monitoring that stuff. I'm seeing from Billy, I assume that you will report significant high water events. Yes. We'll be going on that a little bit more in the restoration takeaways and some of the habitat stuff. But moving on to the, let's see here, 33 minutes. Yeah, I got a little bit. So some takeaways from the restoration experiment. So we've seen great overall lily establishment. Like I said, 25% is pretty great. A lot of restoration projects don't see that, of any, whether plants, animals, don't see that kind of success. So that's cool. We've seen that the wedge technique is most successful and requires about the same amount of person hours as the wrap technique. The difference being that the wrap technique, as well as the mat technique, requires a lot of hard labor, like I was saying, and coir. I purchased a roll of coir, pretty high quality, small hole, like very small holes so they didn't slip out, for my project, with your funding, of course. And I believe it costed $450. So that's not, that cost isn't nothing, or it's something around that ballpark. And on the other hand, the broadcast technique is the easiest, but the success is low and it's difficult to assess. Seedlings appear to be the best restoration life stage, and with bulbs also being very successful. The difference is that seedlings and seeds require significantly less person hours and money. And then regarding season, spring and summer are more suitable than fall for bulb-based restoration, but requires a greater time investment. So based on their life history, if you're going to restore in the fall, you can only use bulbs. And I believe it was 10% success rate with bulbs from the fall. But then if you keep them alive over the winter, invest that extra time and money of electricity and deer defense, as we'll talk about later, but you do get significantly better results if you wait till the spring and the summer. So some more restoration takeaways, and these are more just like based on my experience of being out there and learning from failure and some successes, is you need to be gentle with the roots when removing the propagated bulbs from the pots to limit transplant shock. And I'm sure as a Native Plant Society, you probably knew that, but it's pretty easy to damage the roots when you're taking them out. And then you get what's pictured here in the upper right, this yellowing of the leaves, this kind of burnt look. We had some issues with that, but some of them bounced back, some of them did not. The flow conditions on the day of restoration and post-restoration is important. Now, I can't predict the weather, but it's really important to try to not do restoration right before a big storm. As Bill was alluding to, high water events are the enemy of lily establishment. They have quite the capacity to survive big floods and high flows once they're established. I don't think I have a picture on here, but the creek at the Stevens Creek floods every year often, and in the spring, usually at least a couple times, up on the banks, very high. I don't even know how to describe it. You wouldn't even be able to believe it does it until it does it. So you can't see the lilies, and then the water comes down, and the lilies just pop right up and bloom like nothing happened. But when they're trying to establish, high flows will definitely uproot them and tear them out. So trying to pick the right exact time within a season is important. And you should also be cognizant of the average depth during the growing season of where you're planting. So for example, you don't want to be planting in water that's too deep when it's not even, if you're above the average water depth, or right around the average water depth, and you're planting where it's too deep, when the water gets higher in the growing season, they're going to be underwater. Or conversely, if the water is, you're above average, you don't want to be planting in a dry place, because then they're never going to get water during the growing season. So really kind of having an understanding of how much water is in an area during those key growing season months is important. And then finally, the enemy of spider lilies the white-tailed deer. So deer herbivory is an issue if propagating outside and after outplanting. So we've got game cameras set up all over these sites. We'll talk a little more about that later, but they preferentially choose lilies, especially young lilies, and they will chew them down to the bulb, and which takes a lot of their energy, but can also kill them. So here's a picture of a site area near our site 3 deer in the creek, and it flows pretty fast in here. I've seen them up to almost their bellies in water with the creek just gushing by. So they'll do anything to get a taste of the lilies, unfortunately. So I've been talking a lot, but I'm gonna try to breeze through some of this habitat stuff. And this restoration stuff is a little bit more developed, but the habitat monitoring stuff is, a lot of it's been happening, but there's a lot of analysis that still needs to be done. So I don't have a lot of concrete conclusions yet, but we've been monitoring water quality, water quantity, and herbivory at these sites for, well, before I started, and then through the whole time I've been here too. So we've been using the YSI meter I purchased with your grant funding, and the hobo water level sensors, and a flow probe. And we've also, yeah, like I said, using game cameras. Oh, Bill's talking about post-flood pictures. Appreciate the violence. Yeah, it's pretty wild. When I first started, my advisor and I, because she had never seen the flooding, she thought we were gonna have to change my projects. She took me down there for the first time, and she's like, well, where are the lilies? They're underwater. She just never been down there at that time. She's like, well, maybe they're gonna get washed away. And luckily that has not happened. So we've also, the big piece of my other part of my research is I'm measuring physical habitat characteristics at microhabitat sites within each of these study shoals. And I'm looking at within each of these little 1/2 meter by 1/2 meter microhabitat sites, and looking at water quantity, substrate, exposure characteristics. And I'm going to try to model that, how that relates to the presence or absence of Hymenocallis coronaria. We also, me and my advisor and undergraduate students, kayaked the creek quite a bit, lived out there for a while this summer, and gathered a pretty detailed distribution of where the lilies are within the creek and how many are there. So I'm hoping to compare that with some remote sense data on the geomorphology, hydrology, and solar radiation for each shoal, to kind of see how some like bigger picture characteristics are influencing lily presence and absence at the shoal scale. So some quick and dirty results of some of them from the monitoring I did. We have not seen any ecologically relevant differences in the water quality between the sites. We have observed that pH is increasing in the downstream direction, but it's not at the point that it needs to be an issue ecologically. For example, site 3 is the most basic with a pH of 8.1. But that's very much within the range of a healthy stream still, but still trying to figure out why that is. Surprisingly, we've been using the Clemson Ag Services Lab to analyze our water samples for nutrients, and phosphorus is below the lab detection limit, indicating that the McCormick wastewater treatment plant is doing a better job with their treatment. And then nitrogen appears to be decreasing in the downstream direction, meaning the mill site has the highest nitrogen concentrations, but still significantly below the EPA's standards for what makes a stream impaired. And then looking at water quantity, we've seen that the mill site has the highest discharge, and then site 3, and then site 2, but these are very small differences because there's not very many inputs during that time, tributary inputs. Looking at average channel depth, site 2 is the deepest, and it's significantly deeper than site 1 and 3. So remember, that's the site that doesn't have very many Spiderlilies. So the depth there is interesting to note. And then one of the big picture things I'm working on is creating a site-specific relationship between these parameters, this continuous hobo data. So every 15 minutes, this hobo gives me the water level at a specific location in the creek, and then relating that back to the historic data at a downstream USGS gauge. And then finally, for deer herbivory, we've seen that there's higher deer presence at site 3 compared to site 1. And those cameras have been out there for a long time. And so we've got a lot of data to show that. But we recently deployed cameras at site 2 where we had restoration plots. And there's limited data there so far, but I'm curious to see what we learned there. And there is just substantial physical evidence of herbivory on H. cornaria in the Site 3 area. A lot of stems chewed down and the like. So then this is all preliminary, but some early modeling based on my microhabitat data. I found that water depth and substrate particle size have the greatest effect on Spiderlily presence. So what this means is as water depth decreases, the probability of a lily occurring increases. And as substrate particle size decreases, the probability of a lily occurring increases. And canopy cover and substrate complexity also appear to have an effect, but we need to do some additional modeling and clean up the data before making any concrete claims. But something interesting that we have solidified is this D50, which is the median substrate particle diameter within each shoal. So at the mill, the D50 is 388 millimeters, which is classified as a small boulder technically. And then at site 2 and 3, the D50 falls under bedrock, too big, just continuous bedrock. So that somehow relates to why there are more lilies at the mill than anywhere else. Something about that substrate size is giving more crevices than just a straight bedrock. But I'm hoping to get some more dialing on that quite a bit more. And then the very last thing I got is this is a map of the current distribution of H. coronaria in Stevens Creek. And this is where I hope to use some of that remote sense data to parse out why they're here and where they're not. So you can see at the top of the map, the most upstream is Parks Mill. The first yellow dot is the gigantic colony. And then there's some other little satellite lily populations after that. And then you leave the Parks Mill area, and then there's lilies pretty much spread throughout the rest of the segment, except a gap kind of in the middle where there just aren't very many shoals. But you can see that they're not just at Parks Mill, and there's nothing like the Parks Mill colony anywhere else on the creek. But historically, there were two major other colonies on the creek. One of them has been completely extirpated, and I visited the site. It's just below the bridge that ends my study segment. And then the other major one was at site 3, which is in decline. So it still exists. But what I've seen over my time here is it kind of comes and goes. There are peaks in the lily like any other plant, and then some years like they just don't establish as well, and that's based on what the creek conditions were like during seed dispersal. But I believe that's all on that. I just wanted to acknowledge y'all for supporting my research. And also Clemson Creative Inquiry has provided some funding for travel and additional supplies and undergraduate researchers. The department has been very supportive, and the DNR through my committee member, Mark Scott, has helped a lot with technical expertise. And I'm kind of hoping through this so that I can get them interested in doing a big Spiderlily restoration project, but I'd be lying if I said I'm anywhere close to that. They're like what I'm doing, but I don't think they want to throw any money at it. And with that, I know I just probably blasted you with tons of information. I'm open to take. Say with this beautiful picture of Parks Mill Colony just before sunset.
So how many miles was it in your study section?
Let's see, I work in 8 kilometers. Let's see how many miles that is. I don't know off the top of my head. It's probably like three or four, I don't know. I always do stuff in kilometers. I could convert it for you, but it's 8 kilometers.
Oh, that's about 6 miles.
Okay. And I'm just seeing the chat from Frank about Keith. Yeah, I was specifically not saying any landowners' names because I just out of privacy. But yeah, most of y'all know Keith Self and his wife Becky. That is the location of my third site. Site 3 is the Self's property. So that's the one that has been declining in the last 20, 30 years. but like I said, since I've been around, it's been coming back quite a bit. And I've done quite a bit of restoration there. So I've still yet to see the pictures of how big it used to be. So I don't know if it's gotten back to what it used to be, but it's doing pretty well. There's some very large clumps there, quite a few seeds. There's just nothing at all like this. I mean, there's only one, as far as I know, that in South Carolina, there's only one other place that looks like Parks Mill, and that's Lansford Canal, and Lansford Canal is significantly different as well. But yeah, Keith's site is on the mend.
But Andy, do you have any idea why it declined?
Yeah, that is the $1 million question, and a prevailing opinion was, and I think Frank, you and I may have talked about this, is the increased deer density there, and the potential that there's a hunt lease across. Well, there is a hunt lease, kind of a hunt. What's that called? A hunt club across the creek, and that they're increasing their deer density, and these deer need a place to go, and they're hungry. And based on game camera evidence, there is way more lilies there, but I think it's not, or way more deer, sorry, in that part of the creek. But I think it's more of a combination of factors. The data is hopefully going to bear this out, but Keith's section of creek dries out quicker than the mill does. When there's water, just a little bit of water flowing over the shoals at the mill, most of, or at least half of the channel at Keith's property is dried up. So I think the combination of some of the topography around there allows the water to disappear sooner, and that just invites the deer in there. But I unfortunately don't have my final results on the habitat stuff. I got quite a bit of work in general to do before April, my thesis defense. So I'm hoping to get some conclusions based on that. And it's over Zoom, I may be able to share the Zoom link, and any Native Plant Society members and the like could join in and learn. And I definitely need to send my thesis manuscript to Bill, and then hopefully I hope to have a publication, at least 1 or 2 publications on this that can be circulated around, and you can learn my final results. Or maybe we can do another meeting like this where I join you all again, and I can give you some more concrete stuff related to that.
One other question, then I'll be quiet, but I can't resist asking this one. You know, we've got the dam directly upstream. And one question is, do we think underneath that dam is more of the good habitat that is at Parks Mill? One, and number two, have you had any observations or any inklings about whether removal of the dam would help, hurt, or be inconclusive as to the Parks Mill population?
I would, based on the steep gradient that caused them to build that dam there to begin with, I would suspect that there may be suitable habitat underneath the dam, covered up in a whole heck of a lot of sediment right now. But suitable habitat, I don't think is lacking. There's within that 8 kilometers I'm talking about, there is countless shoals that don't have any lilies or have just a few lilies, and lots of places where you could put lilies. So I don't think we're not lacking habitat in this case. And regarding removing the dam, and I'm hesitant to go fully on record, but right now, my opinion is that I do not think removing the dam would be good for lilies. I think it is providing some sort of mitigation in terms of when big floods happen. I think it reduces the magnitude of the flows compared to further down the stream. Dams also control how deep the water gets just below the dam. So I think it might be helping to keep this just kind of shallow trickle of water flowing over the shoals. But that once again goes back to that all goes back to my hydrology analysis. And once I get some of that hobo data squared away, I'm hoping to something concrete there, because I know you're all very interested in the dam. I would shy away from removing it though. But then again, if it bursts by itself, that's no good for anybody either.
Andy, I would comment that what and it'd be interesting to read some history about how folks sited these dams. Did they waste shoals, which means wasting head by building the dam below where you start having shoals? You know, if I were doing it and I owned the property, I'd build the dam as high up on the shoals run as I could, take advantage of all that head. But
Mhm.
that's just me.
Yeah, that
It'd be interesting
Yeah.
if we could see any history of how these sites were picked. And obviously shows is important.
yeah, I've scanned literature quite a bit for trying to find stuff about this era diversion dams, and there's not a whole lot out there. The best bet might be talking to folks that have any connection to living down there, remembering. I actually, based on a conversation I had with our good friend Gary, he was telling me that the dam there used to be a dam further upstream, and they moved the dam down to where Parks Mill was there. So this is the second dam in that area, as far as I know.
Yeah, Bill Quinn told me that working above the dam for some reason, pulled out wood remains, what looked like a wooden dam.
Yeah, it was wood. It wasn't concrete like this one is,
Right.
too.
So apparently someone put the money into building that concrete dam and just didn't worry about flooding over the old dam. So don't know what to make of that, but
There was a question on chat about any other plants that have these photosynthetic seeds besides Hymenocallis.
So off the top of my head, what I've read is, so it's still Hymenocallis, but almost all the Hymenocallis have this, and in the state, the swamp spiderlily. I'm blanking on I should have practiced my scientific names before I went to talk to Native Plant Society, but I can't remember the scientific name. But the swamp spiderlily has the same photosynthetic seeds, and I imagine that the ones that grow on the coast, in the PD and stuff, the swamp, what is that? The pygmy spiderlily and stuff. I imagine they do as well, but I do not know of any other genuses and stuff that do. But I'm sure that there is. It's a good question. What's this from Catherine? Water Resources Conference. Yeah. Yes, I meant to present at the Water Resources Conference before it was canceled a year ago. And now I believe last time I checked, they are, I guess you put a link here, I can check on that later, but I believe that they put off the next conference until next fall of 23. And I'm going to be, fall of 22, and I'm going to be gone and graduated by then, but that doesn't mean I couldn't still present. I definitely should have some good conclusions by then, so that's a good idea.
Andy, I posted something. I have some photographs from Keith's site from several years back, and I'll scan some of those and shoot you some scanned images. So you can have a feel for what was there before.
Yeah, I would love to do that. I would definitely, I may have a place for that within my thesis as well. Pictures are always great. And yeah. I've heard those exist. I just haven't gotten to see them yet. So that'd be great.
Yeah.
Anybody else got any questions?
Well, I think that Native Plant Society has made a great investment in your work, Andy, and we thank you for reporting to us and got a lot of information on we know a lot more about these plants that we all love now. We thank you very much.
You're here.
Yeah.
We made a great investment.
Yeah, and it hopefully can be an investment that keeps on giving. My advisor probably isn't going to have any more graduate students, but I think she plans to at least keep some sort of presence in terms of undergraduate research down there. Use the equipment we've got, keep the water water quantity monitoring program going forward, and potentially some restoration too. I don't know exactly what her goals are.
I think there might be some interest if we could get some younger folks in here who could scramble around in that creek to do some of that on our own.
Yeah, I will say that, yeah, the doing anything in the creek is a difficult thing. I'm real happy that all my data is collected and all the lilies are put out there, because spending an hour moving around the creek is worth like 8 hours on land. It's stressful.
We
But
saw
nobody
that
got
winter,
hurt.
we saw that wintertime picture of you out there in the creek. So